not? Yes, it is
I
done.
think we have.
Done.
What about types of RNA?
No, we have not done.
So we have to begin from types of RNA today. All right. What happened to your camera? Oh, 1 second. All right. So today we'll begin with talking about the different types of RNA that are there. Now, see, in every individual, there are a variety of RNAs that are present. Okay, there's mRNA, tRNA, rRNA, and then apart from these, there are many different types of RNA also. Like you have miRNA, siRNA, then hnRNA, different RNAs are there. Now, the ones that we have to study are mRNA, where the M stands for messenger. Then after that, there is the rRNA, wherein the R stands for ribosomal. And then lastly, we have to study the tRNA, where the T stands for trans. Understood? So these are the three basic RNA that are found in every individual without any, you can say, exception. Every individual has these three RNA present. So when we talk about the RNA, that is mRNA, so out of the total RNA that is found in a cell, 5% of it is constituted by the mRNA. 5%. That's its, what do you say, population or whatever. And it is the longest RNA, not the largest, longest RNA there is. Other than being called as the messenger RNA, it is also called as the template RNA, nuclear RNA, or informational RNA. Three types, like three names are there for this one particular mRNA. Now, what is the function of this particular RNA? It carries the genetic information that is provided by the DNA. Meaning, you have seen Central Dogma, wherein you saw how DNA, from the DNA, RNA is made through the process of transcription, right? So whatever information is there in the DNA, it gets carried further by the RNA. And which type of RNA is formed after transcription? So mRNA. Understood?
So after DNA undergoes transcription to form RNA, then
Transcription.
yes, ma'am.
Not
After that,
after that, the RNA that is formed is actually the mRNA.
So DNA undergoes transcription to form mRNA. Okay.
Understood? And see, and further, this mRNA only is going to be forming the protein. That is why we say that it is the one that carries the information, the genetic information provided by the DNA, and further up till the protein. Clear?
Yes, ma'am.
Also remember that, do you know what is the meaning of precursor?
The previous?
The previous thing formed before the actual thing, right? So remember that in the case of eukaryotes, when the DNA undergoes the process of transcription, then before the mRNA is formed, there is another RNA that is formed, and that is hnRNA, which stands for heterogeneous nuclear RNA. And this hnRNA, it is the precursor of the mRNA. Got it?
Okay.
The first that is formed, later on that develops into the mRNA. Is it clear?
Yes, ma'am.
Now, moving forward, the next RNA is rRNA. Okay, ribosomal RNA. Now, out of the total RNA in the cell, 80% of it is the ribosomal RNA. Now, this one is smaller than the mRNA, but it is larger than the tRNA. in terms of its size. What does it do? It performs a structural and catalytic role during translation. Meaning, since this is a ribosomal RNA, and you know that the ribosomes are the protein factories of the cell, isn't it? So whenever there will be the formation of protein or anything related to the protein, then these particular rRNA will be coming into play. So ribosomal RNA are the ones that will be forming or that will be performing a catalytic role or structural role during the protein synthesis or protein formation, which we call as, what do we call protein formation as? Translation.
Translation. Oh, okay, okay,
Are
okay.
you able to hear me fine? Like my voice is clear?
Yes.
Now the last RNA, the one again performing like a very major role in the translation process is the tRNA. So out of the total RNA that there is, 15% of it is the tRNA. That stands for transfer RNA, and out of all the RNA, it is the smallest one. And it is also called as the adapter RNA. Now what does it do? It carries or it transfers the amino acid during the process of translation yet again. So mostly the RNA that are there, they are responsible for the formation of proteins all in all. Their major function is to form the proteins. Is it clear?
Yes.
Now we come on to the step that is transcription. Now, if you think about transcription, so this is the process in which the RNA is synthesized from the DNA. Now, obviously, it is a big biological process, just like how replication was. So it would also require a set of enzymes, a proper area, and everything of that sort. So first, we're going to talk about the transcription that takes place in the case of eukaryotes. Sorry, prokaryotes. And then we'll be talking about the eukaryotic transcription. Now see, in the case of prokaryotes, the transcription process, like the site where the transcription happens, it is the cytoplasm. Understood? Now, obviously for the process of transcription, it would be requiring certain enzymes associated with it. So we call those enzymes as the transcripting enzymes. Just like how in DNA replication, you saw that there was the need of DNA-dependent DNA polymerase, right? To make DNA from DNA, that was replication. Now we want to make RNA from DNA. So we require DNA-dependent RNA polymerase. It is still dependent on the DNA because ultimately DNA is the template. But what are we forming now? We're forming the RNA. So we require RNA polymerase. So the major enzyme required during the prokaryotic transcription is the DNA-dependent RNA polymerase. Understood?
Okay, so in prokaryotes, for converting DNA to RNA, they use polymerase for the formation of RNA.
Okay.
We use RNA polymerase for the formation of RNA.
Yes. And we use DNA polymerase for the formation of DNA, like how we saw in replication. Now, this particular enzyme, it's not specific to one type of RNA, that it would transcribe only the mRNA or only the T or only the R. No, it can transcribe all the types of RNA without any exception. Clear?
What do you mean by transcribe?
Transcribe means does the transcription of.
Okay, okay, okay.
Okay. Like how replication replicate, to transcribe, transcription. Understood?
Yes, ma'am.
What are you drinking?
Juice.
Okay, now
It
see
helps.
this RNA. It helps.
It helps me stay awake.
You sleepy?
Yeah, yes, ma'am.
You look tired also, why? Long day.
Yes, ma'am.
And you have your schools, or do you have your summer vacation?
Summer vacation.
Summer vacation. Okay. So why are you tired then?
I have
Tired
to
at
study,
being
right?
home.
I have to study right in the morning.
So see, now this RNA polymerase, it is associated with two factors that will enhance, or you can say that have their own particular role assigned. So there is sigma factor, which is the initiation factor, meaning it will start the process of transcription. It is the initiator. Understood? And there is the rho factor, which is the termination factor. So imagine that the sigma factor came along and now the process of transcription will begin. And then the rho factor came along, now the process of transcription is going to stop. So RNA polymerase has two factors associated with it, which will either initiate the process or stop the process. Understood? Now, the whole entire transcription process, it has been divided into three stages or three steps that are initiation, elongation, and termination. Three stages or three steps are there. Now, during the initiation process, the RNA polymerase, and yeah, one more thing. First, you should know about the transcription unit. See, this is a transcription unit. You remember ORI, origin of replication?
Yes.
That was the point where the DNA replication began, isn't it? Same way, for the transcription to take place, there is a need of transcription unit. And now the transcription unit has two regions. There is a promoter region and there is a terminator region. So the sigma factor of the RNA polymerase, it comes and binds to the promoter region and begins the process of transcription. Whereas the rho factor of the RNA polymerase, it comes and binds to the terminator, thereby stopping the process of transcription. And then you have the template strand, the coding strand, the strands that are normally there in the DNA, the 5' to 3' and the 3' to 5'. So when we talk about the first step, so RNA polymerase, with the help of the sigma factor, it is going to bind to the promoter and it is going to initiate the transcription process. It will begin the transcription process. Then there is elongation, the second step. Now, during the second step, the RNA polymerase, they are going to use nucleoside triphosphate as substrate, sort of like for energy. And they are going to polymerize in a template-dependent fashion. I'll tell you, don't worry. It follows the rule of complementarity. The face, that's why I'm like, wait, I'm here. Now see, what happens is that, for example, this is the DNA segment that needs to get transcribed. Okay, it will obviously have some sort of, what do you call it? What is it, the name of this? Sequence. So there will be sequence present. Like this, anything. It could be anything, random thing. I'm just putting it with whatever comes to my mind. Now, against this DNA, this is a DNA template. So against this, the RNA has to be synthesized, right? So the RNA, that the line says that it polymerizes in a template dependent fashion. Template, this is a DNA template. And according to the DNA template, the new RNA has to be synthesized, right? And it also says that it follows the rule of complementarity. Complementarity means something complement to the strand. So for example, if adenine is there in the case of DNA, right? So complementary to adenine will be present in the RNA. So if I say that this is the RNA, now we have to make the sequences. So according to you, what will be the nitrogenous base that will come in RNA against the adenine?
Cytosine.
Huh?
Adenine, guanine. No. Cytosine, right?
Adenine pairs up with what?
Guanine.
Arey baap re, no.
Once again. Adenine and guanine are pyrimidine and purines, and this is cytosine, uracil,
Give
and
me the whole kahani, but don't give me the right answer. Adenine pairs up with what, Sanjitha? This is very basic.
Yeah. Adenine. And this one,
You're taking the name of all the nitrogenous base except for the one with which adenine pairs up.
The name is not coming out. Once again, cytosine, uracil, and one more.
It's right there in front of you.
Thymine.
Thymine. But
Thymine.
will thymine be here?
No. Yeah? No? You're asking.
Why no? Your parents will think, what are you doing with the teacher that you're laughing out so loud? What will come here? You only have to tell me. Think what we are talking about. This is an RNA.
Thymine. You're
No,
asking.
thymine will not be. Yes.
You're
Yes. So instead of adenine now, it's like normally had it been a DNA, so thymine would be there, right? But since it is an RNA, so uracil. Understood? Now
Yes.
here, what will be there?
Oh God. Thymine. Let's go with cytosine.
Why do you go with it? What pairs up with thymine?
Adenine, uracil. Thymine, guanine.
No.
No.
If adenine pairs up with thymine, then thymine pairs up with adenine. What's so confusing in this? If adenine pairs up with thymine, so thymine pairs up with adenine. It's the same thing. But
Oh yeah.
if adenine is there and you're talking about the sequence of RNA, so you'll not write thymine, instead you'll write uracil. But if thymine is there in DNA, it doesn't have to do anything with RNA. So in the RNA, there will be adenine.
Yes, yes.
Did you get it actually, or just yes, yes, so that I'll keep speaking?
No, no, I understood.
Okay. Guanine pairs up with
Cytosine.
cytosine pairs up with
Guanine.
very good. Then guanine pairs up with
Cytosine.
cytosine pairs up with
Guanine.
adenine pairs up with
Adenine, uracil.
very good. Adenine normally pairs up with thymine, but since we are talking about RNA, pairs up with uracil. Very good. Again, adenine pairs up with
Uracil.
thymine pairs up with
Thymine pairs with adenine.
then guanine pairs up with
Guanine. Guanine. Cytosine.
last one.
C is cytosine. Cytosine pairs with guanine.
This is your sequence of RNA. Now, what have you learned? You've learned that it is happening according to the DNA template. Whatever DNA template sequence will be there, against that a complementary sequence will be made. Clear?
Yes.
And any doubts?
No.
You feel like laughing?
No. I learned what pairs up with what.
Simple. Adenine pairs up with thymine, guanine pairs up with cytosine. Nothing else. If you switch it off, then also it's the same thing. Understood?
Yes.
Now, this RNA is the RNA that has to be made. The DNA will be untouched, unbothered, and the new RNA will be synthesized. Now, remember the helix opens and the whole DNA gets transcribed that way, meaning part by part throughout. Then comes termination. So once the enzyme, that is RNA polymerase, with its Rho factor, when it reaches the terminator region of the transcription unit, now the RNA that is formed, it will fall off along with the RNA polymerase. RNA polymerase is an enzyme, so it will never get used up because enzymes are never used up. They cause the reaction to happen and then they are ready for the next. So this results in the termination of transcription. So here is the picture that tells you about how the process of transcription is taking place, wherein the sigma factor is coming and binding with the sigma factor of the RNA polymerase is binding to the promoter region, thereby causing the initiation, right? And then the elongation happens with the help of nucleoside triphosphates. The RNA, as you see that the RNA, like for example, this is DNA, okay? And this black one is RNA. So as the RNA is slowly getting formed, it is coming out of the DNA helix, isn't it? If you see in the picture, it's coming out as it is being formed. Once the whole entire...
Yeah.
Huh?
Yeah, it is coming
Yeah.
out.
So as it is being formed, it is coming out. Once it's entirely formed, it comes through, like the whole entire RNA strand comes out and the DNA is untouched, ready for the next process. Clear?
Yes, could you show the writings again? I want to read once.
But wait, let me complete. Now see, because we are talking about, very good question coming. Very good question. So the RNA that is formed is the mRNA usually. And further, it requires no modification. Like in eukaryotes, there are further modifications. Imagine detailing of that mRNA happens later on. But in the prokaryotes, no detailing, no modification, nothing. It's done. Understood? Now, after the process of transcription is done, now the next process that has to be done is translation. Now, in the case of bacteria, the process of transcription and the process of translation, it takes place in one common area, that is cytosol, or you can say cytoplasm. That's it. Just both the process takes place in one area only, nowhere else. Can you tell me why?
Now could you scroll a little down?
No.
No. So, yes, a bacteria, why it takes place in cytoplasm? It does not contain nucleus. Nothing. It does not contain
Yes,
nucleus.
I didn't hear. I was drinking water.
It does not contain nucleus.
So
so there's only place cytoplasm.
good. Very good. Exactly, because there's no compartmentalization in the cell of bacteria. There are no membrane-bound structures. So that's why there is only one place for all sorts of activities to occur. Very good. So
That was
also remember, what?
That was luck.
I would have felt better if you would have not told me that. Also remember, many a times in the case of prokaryotes, the process of translation, that is protein synthesis, it starts much before the RNA is fully transcribed. Meaning that RNA is coming out. Now it's not fully out. Like imagine this part is still inside the cell. I mean inside the DNA being synthesized, and this part only is out. So the half of the RNA that is out, that part will start to undergo the process of translation without it being fully transcribed. Okay. Because compartment is same, place is same. So much before it is fully out and ready to for the next process, it starts getting transcribed. Clear?
Yes.
Understood? So the process of transcription in the case of prokaryotic organism is done. Now the next is eukaryotic one. But before that, you note down what is the last thing you were writing?
So, ma'am, initiation, all 3 steps: initiation, elongation,
Achha, you wanted to see. Yeah. See.
RNA polymerase binds to the promoter, which initiates the transcription process. Elongation, the RNA polymerases use nucleoside triphosphate as substrates. It polymerizes in a template-dependent fashion. It follows a rule of complementarity. The helix.
Complementarity.
What do you mean by the RNA polymerase use nucleoside triphosphate as substrate?
That is used, like nucleoside triphosphate is used for the energy for the process to complete. Like it uses that as energy substrate.
Okay, so RNA polymerase uses that as energy. It polymerizes in a template-dependent fashion. It follows the rule of complementarity. The helix opens and the whole DNA gets transcribed. I'm a little down. The whole DNA gets transcribed. Termination. Once the enzyme reaches terminator, the RNA falls off along with the RNA polymerase This is the termination of transcription. Once the enzyme reaches the terminator, the RNA falls off with the RNA polymerase. Once the enzyme reaches the terminator, the RNA falls off along the result is the termination of transcription. The RNA formed is the mRNA. Yes, ma'am.
Any doubts?
No, ma'am.
Anything you want me to repeat?
No, ma'am.
Alright. So what is the last thing you wrote down?
I wrote the question. Why is DNA preferred over RNA? Oh my god.
This is done.
Not write that, the answer. From RNA world. Yes, ma'am.
Done.
No, ma'am.
Okay.
Yes, ma'am. Yes, ma'am. Mom, can I take only these three screenshot and write afterwards?
Okay,
Only these three.
take it now.
Yes, ma'am. Termination. Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am. Can we show that the side one, RNA polymerase, the side one? Yes. Yes, ma'am. Yes, ma'am. Yes, ma'am. Termination, ma'am. Yes, ma'am. Yes, ma'am.
Any doubts up until now?
No, ma'am.
All right. Now, we're going to talk about the transcription that happens in the case of eukaryotic organisms. Now, see, in the case of eukaryotes, the place where the transcription happens and the place where the translation happens, they are very different. In the case of eukaryotes, the transcription, it takes place in the nucleus. Okay. And there are a lot of complexities as such, because you know that eukaryotic organisms, they themselves are complex as compared to the prokaryotes. Now see, in the case of the eukaryotes, what are the complexities? The first thing is that the RNA polymerase, there are different types of RNA polymerases making different types of RNA. Understood? So different polymerase, they make different kinds of RNA. So 3 polymerases are present in the nucleus of the organisms, and they have other polymerases in other cell organelles as well. So when we talk about the first, that is the RNA polymerase 1. So this is responsible for the transcription of 28 sRNA, 18 sRNA, and 5.8 sRNA. Like different S stands for different kinds of RNA. Okay. Then RNA polymerase 3, which will be responsible for the transcription of tRNA. Then 5 srRNA and snRNA, that stands for small nuclear RNA. And last one, that is the RNA polymerase 2. And this one is responsible for the transcription of the HNRNA that will ultimately lead to the formation of mRNA. So that is why we call the HNRNA as the precursor of mRNA. Is it clear?
Yes, ma'am.
Okay. Now see, the basic steps of transcription in the eukaryotes is same just like how it is there in the prokaryotes. But after the transcription is done, the hnRNA that is formed, okay, meaning the precursor RNA, it contains 2 types of sequences in it. The first kind of sequences are exons, and then there are introns. Okay, these are the names of sequences. Now, amongst these sequences, the introns, they are non-functional, mean they don't do anything. They don't code for anything, they don't make anything. So they have to be removed. That is why there is an additional step called as splicing. What is splicing? With the help of certain enzymes, the introns are removed from that hnRNA and the exons are joined together. So now the hnRNA becomes mRNA. Clear?
So after the transcription, before forming the mRNA, the hnRNA formed. The hnRNA just formed contains contains exons and introns. Intron
sequences.
Yes. An intron makes RNA non-functional. So for that, the intron has to be removed. So a special step takes place called splicing, where intron is removed and exons are joined together to form mRNA.
Now we have mRNA, but again, it's not fully processed. So further, 2 additional processing are done, that is capping and tailing. Now in the case of capping, there is methyl guanosine triphosphate, MGTPPT. Okay, methyl guanosine triphosphate. These are added at the 5th, or you can say the 5' end of hnRNA. So at the 5' polarity, methyl guanosine is added and we call it as capping. And at the 3' end, meaning opposite to that, 200 to 300 adenylate residues, meaning adenine residues are added at the 3' end. So this is called as tailing. So capping and tailing are additional post-transcriptional process. Now, the hRNA is fully processed and modified, and now it becomes the mRNA. Got it?
Okay, so before forming the mRNA, the hrNA formed Yeah, the hRNA form contains introns and exons. And next step is the introns. Sorry, ma'am. The introns are removed and the exons are joined together to form mRNA. And again, an additional step, capping and tailing.
No, that
Capping.
that point you can say it is like leading to the formation of mRNA. It is still HNRNA. HNRNA further undergoes capping and tailing. Then it
Okay.
after it
So
becomes
after
fully
this,
processed.
form mRNA again. Before that, there's a capping and tailing where understood, ma'am.
Understood? Now, after the mRNA is formed, now this is formed in the nucleus. Now this mRNA from the nucleus comes out into the cytoplasm for translation. So in the case of eukaryotes, the process of transcription happens in the nucleus and the process of translation happens in the cytoplasm. Clear?
Okay.
And then what happens in the case of this thing, prokaryotes? What happens is that both the processes they happen is the cytoplasm is the location where both the processes will happen. Now the presence of introns is reminiscent of antiquity. This is a phrase that is there in the case of this thing. NCRT. NCRT has this phrase going on. It says that the presence of introns is the reminiscent of antiquity. Antiquity means something that has existed long back, much before the evolution of the genetic material was done. But still it persists. So it is something that is remained even after all these years of evolution. Yeah. That is the presence of introns. Still, in you, in me, in all the animals, wherever the transcription happens, the introns are formed later on, those are removed. Now, the process of splicing, that is the removal of the introns, represents the dominance of RNA world. Again, another thing NCRT says. Why? Because the RNA is deciding to remove those parts from it that are non-functional. And then it is going to keep only those parts that are functional. Clear?
That is RNA is coming out of the nucleus. That's it?
RNA is coming out of the nucleus. No, that is different. See, RNA coming out of the nucleus, that is after it has been fully formed. Now, when it has to undergo the process of translation, then it comes out of the nucleus. Clear?
Yes.
Now, the features of genetic code. Now, see, what happens is that all in all, there are 20 amino acids present. Each of those amino acids, they make the protein. Okay, so the proteins, they are made up of amino acids and 20 amino acids, they have some sort of sequence. Okay, sequence made with A, T, C, and all of that. Okay, so with the help of nitrogenous bases, you have the sequences of amino acids present. Now, when we talk about the sequence, so the sequence that is present in the amino acid, that is called as the genetic code. And there are variety. For example, there is methionine. Okay, methionine is a start codon and it is like it is A, U, G. And always remember, it is always going to be in the form of triplets. Okay, so AUG is the code that it is the codon that codes for methionine. Okay, and as well as it is coding for the start codon. Same way, if you see in your book also, there is this big chart of genetic code given. Yellow color chart. See? See?
See, ma'am, it's there somewhere.
So, there is this chart wherein ATGC, AUG, different codes make up the amino acid. So, that is what we mean by genetic code. So, we are going to see some features of the genetic code. Now, the first feature of the genetic code is that it is in the form of a triplet, meaning three nitrogenous bases are read together, always, never four, never two, never one. It has a commaless nature, meaning that if it is a triplet, then there will be no punctuation in between them, like 3, 3, 3, 3, 3, so on. Genetic code is said to be degenerate. Degenerate means it's not that one code codes for one thing. No, it's that some amino acids they get coded by more than one codon. For example, AUG codes for methionine, like I told you, and it also codes for the start codon. So AUG is a start codon also, and AUG is coding for the amino acid methionine as well.
So it
Understood?
is doing, it's
Two
multifunctioning.
things. Huh. That is why we say that the genetic code is
Multifunctioning.
huh?
You're meaning that it is multifunctional, like it is multifunctional.
Yeah, coding for two things at the same time. So that is why we say that genetic code is degenerate. This property that one codon coding for more than one amino acid, that is degenerate property. Now this is universal. Universal meaning it's triple U is there. U. Okay. So U together, it codes for the amino acid phenylalanine. Now, this phenylalanine is going to be coded with this triplet, that is UUU, starting from bacteria up until human being, meaning in every organism there will be phenylalanine. It will be coded by triple, which is like signifying that it is a universal code.
Okay.
Now, initiator codon or start codon, like I told you, that AUG, it is coding for methionine as well as it is a start codon or initiator codon. Then stop codons. Now these are very important. Like these are the ones that if they are going to be present in any of the sequences. Okay, what are they? UAA, UAG, and UGA. Three stop codons or terminator codons are present. UAA is named as ochre or ochre, whatever. It's on the name of a person who discovered it. Amber, I should write it with capital. You write it with capital letters. So amber, UAG is the stop codon and opal, UGA. So they don't, like these are the codons that are not coding for any amino acid. So we call them as nonsense codons because they don't code for anything, but they are the stop codons. For example, like how AUG is there. So from here, any process will begin, whether it is transcription or anything. UUU, like translation of this will happen and the phenylalanine will be formed and like that. But wherever the UGA, that is a stop codon will be present, at that position, the whole entire process will stop. It will not further go on. Understood?
Yes, Mama. What is A? A is adenine. U is it's like that.
Huh.
U is uracil. G is guanine. Okay.
So genetic code is clear.
Yes, ma'am.
Oh, now look up. Transcription in eukaryotes is clear to you?
Yes. Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am. Fusion.